In this presentation, we will be looking at the importance of chloride ions in the central nervous system and skeletal and smooth muscle. We will cover 3 specific topics of interest. The first topic that we will address is how GABA exerts its physiological effect and how it affects neuronal excitability in the central nervous system. Gamma-aminobutyric acid, or abbreviated as GABA, is an amino acid and the main inhibitory neurotransmitter in the brain. GABA neurotransmitters can be further categorized as GABA A, GABA B, or GABA C. In this presentation, we will focus primarily on GABA A. As you can see in this figure, GABA neurotransmitters are released from the presynaptic neuron into the cleft during exocytosis. Once the GABA vesicles reach the postsynaptic membrane and bind to GABA receptors, ligand-gated receptor channels become stimulated and change shape, then permitting the inflow of chloride ions. As you can see, this inflow of the negatively charged chloride ions reduces the charge within the postsynaptic cell, causing hyperpolarization, making it harder to reach threshold, hence having an inhibitory effect on the postsynaptic cell. The next topic discusses benzodiazepines and their effect on neural excitability and why the combination of Valium and alcohol, or barbiturates, is worse than taking Valium alone. Benzodiazepines, such as Valium, are a therapeutic drug that can relieve symptoms of anxiety by enhancing the inhibitory effect of GABA on the brain. As you can see in this figure of a postsynaptic membrane, benzodiazepine or other barbiturates has the ability to change the shape of the primary ligand channel. The benzodiazepines enhance GABA, opening the chloride channel wider and for an extended period. This conformational change causes a greater inflow of chloride ions, which in turn lead to hyperpolarization and prolonged inhibition of the postsynaptic neuron. Alcohol also acts similarly to benzodiazepine in that it potentiates or enhances the effect of GABA. As a result, the ligand-gated chloride channels become more permeable to chloride ions, also producing an inhibitory effect. When combining a drug such as Valium with alcohol, there can be high-impacting or fatal consequences. According to studies by Tanaka, in general, metabolism of these drugs is delayed when combined with acute alcohol ingestion. Alternately, some reports show the drugs metabolized during chronic alcohol ingestion have an increased clearance. The third topic that we will cover today discusses the physiological function and movement of chloride ions in skeletal muscle and the channelopathy myotonia congenita. In skeletal muscle, the major channels responsible for the movement of chloride ions and recovering resting membrane potential are voltage-dependent anion channels known as the isoform CIC1. Chloride ions act as a ligand to their own channel as it controls when and what passes through each channel. Channelopathies are diseases caused by impaired channel function from channel gene mutations. Myotonia is a condition where patients experience delays in muscle relaxation after contraction. In the regulation of muscle and neuron excitability, chloride, as an electronegative ion, plays a vital role. Chloride ions, alongside intracellular and extracellular pH levels, also modulate channel function and depolarization. As illustrated in this figure, in regular muscle fibers, dihydropyridine L-type calcium channels, annotated as DHPR, and in turn intracellular ryanide receptor channels, annotated as RYR in this figure, open from stimulation of action potentials. Muscle contraction occurs when calcium levels in the sarcoplasmic reticulum increase from the opening of these channels. Repolarization of the membrane then occurs as the potassium ions move out of the cell. After the repolarization, small depolarizations may occur in the T tubules from the accumulation of potassium. Prevention of depolarization propagating along the sarcolemma is mediated by chloride ion conductance of the CIC1 channel. In contrast, CIC1 channels in the muscle fibers of patients suffering from myotonia lack this conductance of chloride ions, which eventuates to autonomous sarcolemmal action potentials, causing delayed relaxation of the muscle. Thank you for listening to this presentation on the importance of chloride ions in the central nervous system and skeletal and smooth muscle.